Hey everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to talk about another paper in the story of kinesin, titled Kinesin is bound with high affinity to squid axon organelles that move to the plus end of microtubules. It was written by Bruce Schnapp, Tom Rees, and Ruth Bethtoel, and it was published in the Journal of Cell Biology in 1992. In our last lecture, we said that you could extrude the cytoplasm, or axoplasm if you prefer, from the squid giant axon, the cytoplasm of the axon, the axoplasm. And you can separate out different components by first adding a very strong salt, potassium iodide, that breaks some of the weaker bonds between proteins and other components of the cell. And if you add the axoplasm up here to a 12%, 15%, 45% sucrose gradient, then the axoplasm will float on the surface until you centrifuge it under high force. And when you do that, the vesicles end up in this 15% layer. And in this paper that we're talking about today, Bruce refers to the vesicles as organelles, which are both valid terms. They are small spheres that have a lipid bilayer. That is, it's like a basketball where the cover of the basketball is lipid bilayer membrane. And of course, the basketball is filled with air, and these vesicles, or organelles if you will, are filled with cellular components, proteins and other things. And you can also have up here you'll have things that are soluble in the supernatant, that are light, that are small and light, and they're soluble. And of course you can have things in the 12% layer, indeed you do, and in the 45% layer, and even stuff that's pelleted to the bottom of the tube, like this. But we said that if you take the supernatant, the S1 fraction, and you put a little droplet on the slide and then remove the excess, so you just have a thin layer of the supernatant on the slide, and you add microtubules and a little bit of buffer solution so that it's in a solution, and you add some ATP, that the S1 fraction can cause the microtubules to glide or slide along the cover slip within the area of the droplet, which demonstrated that the kinesin, the motor protein that was in the supernatant was attached to the glass and moving the microtubule along like a beach ball at a rock concert. And it turns out if you biochemically purify kinesin, which Ron and his colleagues did, very famous experiment. If you have purified kinesin, kinesin will also facilitate microtubule gliding along a glass cover slip. And if you put microtubules down first, the microtubules on the glass, then the microtubules bind to the glass through a charged interaction, and if you add the supernatant or kinesin to latex beads, that is, soak the latex bead in the S1 fraction or kinesin, then the glass beads move along the microtubule. And they do that at a rate of 0.5 micrometers per second. And we said then, if you take the organelles, as Bruce calls them, or vesicles, and you add those to microtubules in the presence of ATP, the organelles or vesicles also move along the microtubules, but they move at about 1.6 micrometers per second. Okay, then we said if you take both the latex beads and the organelles, there's a latex bead that's been soaked either in kinesin or in the S1 fraction, and you have organelles, they both move in the same direction, just at different rates. And then if you take spindles from the centrosome, like this, and you add the latex bead soaked with S1 or purified vesicles, they both move in the same direction, and they move in the direction from the MTOC, the microtubule organization center, away. And we know from other experiments that the plus end of the microtubules in centrosome is away from the MTOC. This is the minus end, and that's the plus end. So kinesin causes the latex beads to move in the plus direction, and the organelles, or vesicles if you will, also move in the plus end direction. So now from what we've learned about the polarity of microtubules in an axon, we said, based on Bass et al., that the microtubules are in the axon here. There's our nucleus and, of course, our dendrites here. And we said that in the axon, all microtubules are oriented with the plus end away from the cell body and the minus end towards the cell body. So kinesin causes, in this case, latex beads, if you want to say it that way, but it promotes transport towards the plus end of the microtubule. It turns out that from Ron's work with video-enhanced DIC light microscopy and extruded exoplasm that's been diluted, he showed that single microtubules can support bidirectional transport. So there are vesicles or organelles that also move in this direction. And we're not going to talk about it, but there's another set of really wonderful experiments that suggest that another protein family called dyneins is responsible for transport in this direction. So this model was set up that kinesin moves things down the axon away from the cell body towards the plus ends of microtubules, while cytoplasmic dynein moves vesicles in the opposite direction towards the negative end of the microtubule and towards the cell body. The problem is that there's no evidence that links the motor in the supernatant or kinesin with the vesicles. And the fact that the organelles or the vesicles move at 1.6 microns per second, while kinesin and the S1 causes latex beads to move at 0.5 microns per second. Of course, one explanation that I mentioned the other day is that potentially in the process of biochemically purifying kinesin or separating out the different components of the axoplasm in this sucrose density gradient somehow damages the motor. And that's certainly a valid explanation. But at any rate, this discrepancy stood. I mentioned earlier that in 1985 I began working as a scuba diver for the Marine Biological Laboratory in Woods Hole, which is the same institution where Ron Vale, Tom Rees, Bruce Schnapp, and Mike Sheets did their experiments on axonal transport. And in fact, in 1985, that was the year that Ron and his colleagues biochemically purified kinesin. At that time, I didn't know anything about axonal transport, but in 1986, I was hired by Tom Rees to dissect squid optic lobes and axons to support their experiments. And I met Ron, Bruce, Tom, and Mike that year. A few years later, after Ron had discovered kinesin in squid, he was interested in trying to figure out whether kinesin might have a role in cellular division, and I ended up scuba diving and collecting surf clams for Ron. The fertilized eggs from the clam were used as a model to understand how cells divide. Bruce, who was a postdoc for Tom Rees in the time of the discovery of kinesin, ended up getting a faculty position at Boston University and then at Harvard Medical School. By this time, I was very interested in the axonal transport story and working on kinesins in general, and I asked Bruce for a job, and so I worked for Bruce at Harvard Medical School for a year during the time they were working on the publication that we're talking about today. My project in Bruce's lab was to make an antibody against kinesin so that we could do some really cool downstream experiments. I know you guys know a lot about the immune system, but let me just mention a few things. This is my bunny. And there are a number of ways to create antigens. We know that when a foreign substance enters the body, there's an immune response, and part of that immune response is the generation of antibodies, which are proteins. So they're generated by genes, and protein looks like a Y, and it turns out that most of the protein is identical. Every single one is identical. So if you make an antibody against influenza, and then your body makes another antibody against corona, for instance, then this part of the antibody is the same. They call that the conserved domain. And it's the little tips that are called the variable domains that are specifically made to that particular antigen. So it would bind to influenza, for instance, in a lock and key type of mechanism. And this variable domain would be different if the antigen or the infection is something else like coronavirus. There are three typical ways to make protein antigens. The first is, just simply, I don't know if it's simple, but biochemically purify whatever substance you want, like kinesin, for instance. So you could take biochemically purified kinesin and inject it into the rabbit and generate an immune response. And then once the antibodies are developed by the rabbit, you can bleed the rabbit, take some of the blood from the rabbit, and you spin down the red blood cells and lymphocytes, and what's left is the supernatant, and that's enriched in the antibodies. So you have a solution now that contains high concentration of antibodies. And of course they did do this. They took kinesin and injected it into a rabbit, but they were running out of this antibody, and so Bruce wanted me to try to make a second antibody. So we could have gone back to the well and biochemically purified kinesin. In fact, I did purify kinesin with Bruce, but we decided to make a different type of antigen. Now this is the first way. If your antigen is going to be a protein, you could just biochemically purify it. Second, if you know the amino acid sequence, which by this time we did know, then you can look at the amino acid sequence, like here's the N-terminal and the C-terminal of the protein, and you can look for a region that you think might be antigenic. Maybe an area where there are amino acids that have a lot of side chains, and maybe that makes it a little bit more recognizable as foreign by the rabbit. So they look at the total amino acid sequence, and they just pick a region that looks promising, and they generate that in a test tube. You can make a short stretch of amino acids with a machine, and then you can inject that into the rabbit. And so they call that a peptide antibody. Peptide, because you're just injecting a short peptide, not the whole protein here, like the purified kinesin. And the third way of doing it is to overexpress the protein in bacteria which they call, recombinant. So sometimes you'll see, if they're in the literature, when they're talking about recombinant antibodies, they put a little R for recombinant and then kinesin, so that you know that the kinesin was generated by growing it up in bacteria, by synthesizing it in bacteria. So what we knew first was, of course, we know the full-length amino acid sequence, but that was derived from knowing the full-length RNA molecule. So this guy, Ken Kosick, who now works on Alzheimer's disease, figured out the cDNA sequence of the RNA that encodes kinesin, conventional kinesin, Ron's kinesin. So, of course, we said this before. So you take RNA molecules and then you add a string of T's that bind to the A's, and then you add reverse transcriptase, which reads whatever nucleotide is here, like a G is up here and it makes a C, and if it's an A up here, it's a T, and so on. You guys know that. And now you generate a complementary strand to the RNA molecule. But this is DNA. So you have a DNA RNA hybrid at this point, and then you add an RNase, which chews up the RNA. You add DNA polymerase, and now it reads the complementary strand, and it makes a complement of the complement and rebuilds this strand here. But now the U's that were in the RNA are replaced by T's, and you end up with a double-stranded version of the double-stranded DNA version of the original RNA molecule. And we said that when you're looking at the RNA molecule, the entire RNA does not encode amino acids. Only a portion of the RNA molecule encodes the amino acid sequence. And so there is a start site. They usually draw a little arrow like this. That's the start, and that encodes methionine, because all proteins begin with methionine. And then there's a stop codon somewhere down here where the ribosome stops reading the RNA sequence and stops synthesizing the protein. But at any rate, you would synthesize a protein like this, from the start site to the stop codon. And then this protein, of course, folds up into a three-dimensional structure, which looks like this. That's the kinesin, and it turns out that a mature kinesin molecule isn't made of one copy, but two. It's a homodimer, and so their tails wrap around one another. And then these heads, as we've said, walk along the microtubule. So it would have been nicer if we had called them feet instead of heads. But at any rate, there are also some light chains, some smaller proteins, accessory proteins that are bound to the kinesin molecule. So what Bruce wanted to do was make an antibody just against the head domain. So instead of PCR amplifying this full stretch, we made a PCR primer, one that would bind here near the start site, and one that would bind, part way down the RNA molecule. And if we convert this to cDNA and then use these two primers in a PCR reaction, we would generate, a piece of RNA, double-stranded DNA that's the copy of that stretch of the RNA molecule, and it would encode just a piece of the kinesin protein, the head. That's what Bruce wanted. So to make it in bacteria naturally have these circular pieces of DNA called plasmids, and scientists have genetically engineered the plasmids so that you can take a piece like this generated from a PCR reaction and ligate it into the plasmid and recircularize the plasmid like this. And then you can insert that into a bacteria like this, and you can turn this gene on. The part that you inserted into the plasmid gets expressed, and it ultimately makes protein. It's RNA and then protein. So now you've got a bunch of kinesin heads in the bacteria, and they make a little tail. They add a few more amino acids here that they know will bind to nickel in a column. So now you can have beads with nickel on them, And if you grind up the bacteria and you pour this over the column, the heads, the kinesin heads with the little tails stick, and everything else gets washed through. All of the bacterial cells get washed through, and then after it's all squeaky clean and it's just the beads and the kinesin heads, you can change the pH and you end up getting a vial of recombinant kinesin heads. And of course, you could have made an entire recombinant protein if you wanted to, but we just wanted an antibody that will recognize these heads, and so the heads end up being injected into the rabbit, and the rabbit makes the antibodies, and then we recover those antibodies from the blood of the organism in the supernatant after you spin down the cellular debris. Okay, so just a couple other notes. First of all, I mentioned that they add a few nucleotides here at the end, and those nucleotides encode histidine, the amino acid histidine. And if you have a stretch of about 6 or so, they have side chains that somehow interact with the nickel. So you can use that to generate this affinity purification column to purify the recombinant protein. So it's a polyhistidine tail. The second thing I wanted to mention is that the coronavirus vaccine from Moderna and Pfizer are not protein antigens. They're a piece of the RNA molecule that's encoded by the virus, and that RNA encodes one of the proteins that is expressed on the surface of the virus itself. So the vaccine that we're getting is an RNA vaccine, and that's being injected into us, and then our body is making the protein from that RNA, and the protein then becomes the antigen. So ultimately, the antigen is a protein again, but the vaccine itself is an RNA vaccine. Okay, so there are many experiments that were done in Bruce's paper, but I'm only going to talk about one that's in the paper that worked, and one experiment that I did that didn't work. So the figure I'd like to talk about is figure 4, which is a Western blot. So in a Western blot, you take a sample that you're interested in. In this case, we're going to talk about our sucrose density gradient one more time. So we have our test tube, and we have it filled with different layers of sucrose, 12%, 15%, and 45%, and we've taken some axoplasm and diluted the sample and added salt, potassium iodide, to break things up a little bit, and loaded it onto the top. And then we centrifuged the sample, and as we've said before, soluble factors, including kinesin, is found in the supernatant, and then we have our organelles or vesicles, whichever you want to call them, down here, okay? And in figure 4, Bruce has a Western blot, and what you do in a Western is, first of all, you run the protein out on a gel. So protein gels are a little different than DNA gels because they stand up. They're vertical gels, but there's a couple of glass plates, and there's in between the glass plate is acrylamide. So that's the matrix. It's a little different. It's agarose for DNA and RNA gels, and acrylamide for protein gels. And so there's a couple of sheets of paper, a couple of sheets of glass, and there's like a couple of millimeters of acrylamide, but they have the little wells in the acrylamide. So this here is acrylamide, and all the rest of the gel is acrylamide, but they have these little wells where you can load your sample. So you load your sample in up here, which contains a whole bunch of proteins. It's a protein gel, so that's what we're looking at. But of course, it's everything. It's everything that you put in there. If you just grind up brain tissue, it's not just proteins. There's lipids and other compounds in there. But at any rate, the proteins, the sample is loaded in here into what they call the well, and then you apply electricity, and the electricity runs in this direction, and the proteins migrate through the acrylamide based on their molecular weight, by their size. The small stuff travels faster because it's smaller and it can get through the matrix a little bit easier, and then the larger proteins are slower. So if you ran the protein gel of something like the S1 fraction, and then you'd have to stain it because you can't see the proteins unless they're stained. It would just look clear if you run the gel without staining it, but you run the gel and then you throw it in some blue stain, Kumasi stain, and then you would get a banding pattern for all of the different proteins, like we talked about in the purification of the acetylcholine receptor. Then once you have your gel run, so that if it's not stained, then you just have the gel and you pop it out of the plates. So you have these wells like this now. And something like this, okay? And of course, you would have the banding pattern here, but it's invisible because you're not staining it in this particular case because you're going to do a Western blot. And then, after the gel's completed, you lay it on a piece of fancy paper, nitrocellulose, and you run the electricity now through the gel. So it transfers the proteins to the piece of paper. So now you have a piece of paper, and you could stain the piece of paper. And if you stain the piece of paper, you'd see the banding pattern like this. But we're not going to stain the Western blot. We're going to use an antibody now against kinesin and see whether kinesin is in the sample. I mean, it's a test to figure out whether kinesin is in this sample and whether it is in this sample, because Bruce is trying to figure out whether the organelles or the vesicles that move to the plus ends of microtubules is driven by conventional kinesin, by the kinesin that he and Ron and Tom and Mike worked on and purified from squid optic lobe. We know these move, but did they move because of kinesin? Okay, so what he does is he runs some of this in one lane, and he calls this cytosol. Cytosol. So he instead of calling it the S1, he calls it the cytosol, which is fine. He just uses a different term. And then now he calls these, loads these into the second lane, and now he calls what he was calling organelles in the title, he calls Ki vesicles. Okay, so Ki vesicles. But like I said many times before, they're just interchangeable terms. And then you run both on the gel, and then you transfer it to the piece of paper, and now you have your membrane, and you have two lanes. You have lane 1, which is cytosol, and you have lane 2, which are the vesicles, right? And then he incubates this piece of paper with the kinesin antibody. And the kinesin antibody, of course, binds to kinesin like a lock and key sort of mechanism. So if there is no kinesin here, well, it's not going to bind. But we know that there's kinesin in this cytosol already because they've used antibodies and shown that the protein that's in the S1 or the cytosol is kinesin, is the kinesin that they purified from squid optic lobe. So we know that there's definitely original conventional kinesin in the cytosol. So there's a bunch of proteins here, and if you have kinesin here, then the antibody is going to bind to it like lock and key, right? And they add a second antibody that recognizes the first antibody. So some company took antibodies from rabbit and injected it into another organism, like a donkey. And the donkey recognizes the rabbit antibody as foreign, and the donkey now builds donkey anti-rabbit antibodies. It has donkey antibodies that will bind to rabbit antibodies. And the kinesin antibody was generated in rabbit. So the company then takes the antibodies from the donkey and they conjugate it to different things that can be detected. So they could conjugate that antibody to something like green fluorescent protein if you wanted to look at a sample in the fluorescent microscope, so you need a fluorescent tag. If you want to look at it in the electron microscope, they conjugate that second antibody to a piece of gold, a little gold particle. And in this case, they conjugated it to something that gives off light. And you have to, after you put the primary antibody on and secondary antibody on, you have to put a piece of film, photographic film, over the piece of paper, over the nitrocellulose, of course in a dark room. And the second antibody gives off light, and then when you develop the piece of film, you can see the band on the piece of film. So it's called chemiluminescence. So of course you could conjugate the fluorescent molecule directly to the kinesin antibody, but it's just more convenient if you have a whole variety of secondary antibodies that bind to the first that will allow you to use the kinesin antibody for a Western blot, for confocal imaging or electron microscopy and so on. It's just for convenience, so they use two different antibodies, one that binds to the target, in this case kinesin, and the second one that's conjugated to some sort of tag, some sort of detectable marker, binds to the first. It's just simpler that way, easier to work with. Okay, back to our Western blot. So now they have detected a band for kinesin in the cytosol, which isn't a huge find because they already knew that kinesin was in the cytosol. The question is kinesin in the vesicle or organelle fraction? Because Bruce is trying to figure out whether kinesin, that is the kinesin that they biochemically purified and the kinesin that is found in the cytosol, is it attached to the vesicles and found in this fraction? So the only way that the molecule can get all the way down here into the 15% layer is if it's bound to something that drags it down into this layer, because if it's by itself, it's just soluble and it's going to stay floating up here in the supernatant. But at any rate, that's what they want to test, is what happens if you load these vesicles on. And if you load the vesicles on, they get a band. So they have two bands. So okay, there's some amount of kinesin in the organelle fraction, a detectable amount. They can detect it here. The interesting part of the paper, for me, is that Bruce tells you in the figure legend that the cytosol is diluted 1 to 1,000. Right? So if this volume of the vesicle fraction or organelle fraction and the volume of the cytosol is the same volume, and you add the same amount of each one to lane 1 and lane 2, and the two bands are equal, then you would say that the protein is of the same concentration in both samples. I mean, the same volume, you added the same amount, and you got the same size band, and so they're equal in concentration. But Bruce tells you that he's diluting the cytosol fraction, this fraction, 1 to 1,000. And so if you load this very, very dilute cytosol on here, 1/1000th of what you're loading on here, then, and you get the same size band, it means that there's 1,000 times more kinesin in the cytosol than there is in the organelle fraction. So it means that there's only 0.1% of kinesin that is tightly bound to the organelles and making it into the organelle fraction. So, of course, it was detected. I'm not arguing that it wasn't detected. It is detected. But it's just there's such a small amount of kinesin in the vesicle fraction that I'm not sure that would account for the transport. So that's part of the problem. So that experiment, I guess, worked. But what it demonstrates to me is that there's a very, very tiny amount of kinesin that made it into the vesicle fraction, and it's probably a contaminant. It's just too small of an amount. Really, kinesin is not in the organelle fraction. 99.9% of the kinesin is in the cytosol. It's in the supernatant. So my experiment for my project in Bruce's lab was to take the vesicles, which we know contain a motor for sure because they move towards the plus ends of microtubules at 1.6 micrometers per second. And as we've said, this cytosol moves latex beads at 0.5 microns per second. And if kinesin is attached to these vesicles, then you should be able to take the vesicles and put them on a slide. This is a slide for an electron microscope, which is called a grid. Actually, the grids are circular. Maybe I should draw it like that. But if you put the vesicles now on the surface of the grid, and you add the kinesin antibody, the kinesin antibody should find the kinesin on the surface of the vesicle and bind to it. And then if you add a second antibody that binds to the first that has a gold particle attached to it, because we're going to use an electron microscope, so you can see a gold, it's called colloidal gold, but it's a little tiny gold particle, and it's electron dense. It shows up as a black dot. So you should see a vesicle, this, if it has the kinesin, like this, then the primary antibody should bind to it like this, and then the secondary antibody, which is conjugated to a little piece of gold, would show up like this. And you wouldn't be able to see the kinesin in the electron microscope. What you would see is this is the vesicle, and then if there's a kinesin there, that you would see a gold particle. And if there's multiple kinesins there, then you'd see a bunch of kinesin particles like this. Okay? So at any rate, my experiment didn't work. It didn't work. It didn't work because there's very, very little, if any, conventional kinesin in this fraction. The kinesin is in the supernatant. Okay, you guys, that's it for today. See you next time.